Good morning. So today I have the distinct pleasure of welcoming Dr. Alan Remaley, today's speaker, a friend, colleague, and a collaborator, and a wonderful researcher. So Alan got his MD Ph.D. at University of Pittsburgh School of Medicine, and he was trained as a clinical pathologist at UPenn. And then he went to NIH, and so he comes to us from NIH, where he is the section chief on lipoprotein metabolism at NHLBI. And at the same time, he also has service responsibilities in the Department of Anatomical Pathology, Director of Special Chemistry. So while Alan has significant service responsibilities, you probably wouldn't know if you followed him as a researcher, because he's so productive in both basic and translational research that he's just more productive than most people that I know who's doing full-time research. Alan is a leader in an area of HDL research, published contributed his contribution in this area. So really too many for me to talk about. He's, among other things, also editor of one of the editors, editorial board of Ph.D. Now just when we think that we know everything about HDL, so-called good cholesterol, there has been some controversies on HDL because of partly because big pharmas spend billions of dollars in producing drugs that raises HDL, but don't seem to prevent CHD. So this is becoming again very, very interesting, very hot area, and Alan being one of the leaders in that. So I invited Alan here 4 or 5 years ago, and he gave one of the talk updates on HDL. But since then, his lab has come up with a lot of new information, and I thought I'd invite him, and he kindly agreed to give us an update on his research.
Mike, is the microphone on?
Oh, I'm sorry, I don't know. So I hope most of you heard some of what I said.
Can everyone hear me?
Yeah.
Yes. Great. Well, thanks, Mike. It was a very kind introduction. And Mike and I have been friends and collaborators for many years. And Mike is clearly one of the leaders in cardiovascular biomarkers. And as Mike said, I was here. Actually, I looked up in Google because it was after your bridge collapse in 2007. So I was a little bit worried about coming back, whether there were some other disasters. But you had that freakish hot weather yesterday, which was quite remarkable. But other than that, it was fine. So when I was here, I guess in 2007, I talked at the time about Transient disease and LCAT deficiency, mostly about discovery of genetic defects and some basic biochemistry. And what I'm going to do today is talk about what we've been doing the last few years, trying to translate this knowledge into 2 new therapies for cardiovascular disease. So just under disclosure, so some of the work I'm going to talk about, we've had and we licensed to AstraZeneca and Kinemed, and I have grants from both these companies for working on these compounds. So I'm going to give everyone a crash course in HDL biology, so everyone can understand what I'm going to present. Then I'll talk about my work on lipoprotein peptides, and then a key enzyme in HDL metabolism called LCAT, lectin cholesterol acetyltransferase, that esterifies cholesterol. So there's 2 kinds of HDL in terms of release shape, and the newly synthesized HDL is essentially a phospholipid biolayer structure. This is produced in the liver and to the small degree the intestine. And the acyl chains, which are very hydrophobic, are kind of protected by the amphipathic helices on ApoA1 and the other proteins around HDL. So this is called pre-beta HDL based on its electrophoresis migration. And it's one of the initial receptors cholesterol from cells. And the predominant form of HDL is actually a micellar structure, so it's a single layer of phospholipids. And the cargo is inside, and cargo in case of HDL is mostly cholesterol ester and triglyceride. And this is the predominant form, and this is the form that after it removes cholesterol from cells, it delivers the cholesterol to the liver. And LCAT is an enzyme that esterifies cholesterol, and I'll go into greater detail, but it converts the disc-shaped HDL into the cholesterol, ester-containing HDL by esterifying cholesterol. So the pathway that I study is called the reverse cholesterol transport pathway, and this is the pathway which the body maintains its cholesterol homeostasis. So cholesterol, except for a few tissues, cannot be further catabolized, so it tends to accumulate, and the way you maintain homeostasis is through HDL removing excess cholesterol. So the pathway begins when apoA1, the main protein component of HDL, is produced in the liver and acquires phospholipid cholesterol and forms this discoidal structure that I just showed you a second ago. It then goes out to the periphery where it acquires additional cholesterol by other ADC transporters and forms what's called alpha-4, another larger discoidal structure. And then finally, LCAT, the enzyme, esterifies cholesterol, and it goes into the core, converts this into the sphere. It's not trapped on HDL until it can be removed either directly by the liver or it's transferred to LDL by CETP. And I'll just talk a little about this later on, but this is why inhibition of CETP has always been controversial. If you block CETP, you increase the cholesterol content on HDL, but you potentially block the whole pathway for its return to the liver. So, it's important to realize that the whole size of HDL cholesterol doesn't tell you about the flux. So if the benefit of HDL is the flux, the CETP inhibition may not be useful. So I'll talk today about apoA1 mimetic peptides and LCAT, two ways that intervene in this pathway. So most of what I'll talk about is cholesterol efflux, this is shown here. But HDL has many other properties. It has about 80 proteins associated with HDL. It has about 20 or more different bioactive single lipids. So it has potent anti-inflammatory effects on monocytes, on endothelial cells. HDL also has peroxidase and several enzymes that break down and oxidize lipids. So it prevents the oxidation of LDL, which is thought to be important in the initial generation of macrophage foam cells. And I'm going to show you data that HDL can remove cholesterol, but it can actually remove as much as 50% of the cholesterol in the plaque with just a few infusions. Part of that is due to cholesterol leaving the cell, but also these cells, they become immotile. And once you remove a little bit of the cholesterol, they express different chemokine receptors and actually very quickly migrate out of the plaque. Now, as Mike said, there's been great interest in HDL for this reason. So even though I think statins are wonderful drugs, under the best of circumstances, they only reduce cardiovascular events by about 30%. So those are compliant patients in clinical trials. The reality, patients aren't compliant, they don't take their medications, or like former President Clinton, they stop after 5 years and feel better. So we're not doing as much good as we think we are. Now there's several small studies showing that if you lower LDL and raise HDL, and most of these studies are done with niacin, you can maybe reduce events 80 to 90%. But many of you may know there's been recent studies questioning whether niacin indeed works or not. And the other way to raise HDL, as I mentioned, was a CETP inhibitor, and there was a spectacular failure by Pfizer a few years ago now showing that torcetrapib, which raised HDL cholesterol, actually increased cardiovascular events, although it's probably off-target effects. And then just last year, another CETP inhibitor from Roche, delcetrapib, did not increase cardiovascular events, but was halted because of futility. And there's still 2 other CETP inhibitors that are being evaluated. Now, what I'm going to talk about today is trying to address this problem. So this is the frequency of recurrent cardiovascular events in patients with acute coronary syndrome. So these are patients who present to the ER with myocardial infarction or stable angina. And when I was in medical school, it was much worse. Now it's not as bad, but still quite bad. That you see you have about a 15% chance of having another event in a year period, and usually half of myocardial infarctions are fatal. If you go out, it goes out to about 25% in 2 years. Now, you start patients on beta blockers, antiplatelet therapy, you start them on statins, but you still have this acute rapid increase in events, and statins don't really start having benefit after about 4 to 6 months. So people are interested in trying to use HDL to address this problem. And I thought this was a crazy idea. This was an idea developed by Roger Newton, who developed Lipoprotein, and he started a company called Esperion. And he used a natural mutation in APOA called A1 Milano, which is RH assist substitution. It's thought that this free sulfide group makes it better antioxidant, and it's associated with longevity. And I was skeptical, but the animal data was very persuasive, showing that just a few infusions of HDL, intravenous infusion, rapidly reduces inflammation and plaque size. And there's now several clinical trials. These are IVA studies, or intravascular ultrasound, where they look at the plaque. These patients are usually treated once a week for 4 to 5 weeks with a very high concentration, about 45 mg per kg of APOA1, which is the main protein component combined with phospholipid. So it's a very expensive treatment. These patients typically receive about 3 grams of APOA1 per treatment. And what they found is that they see a reduction in plaque size equivalent to about 2 years of statin treatment. So there's great interest whether this will translate into cardiovascular protection. But it's been difficult to produce the quantities of APOA1 needed for the study. So this is why I was interested in whether one can use short peptide mimics of APOA1 as alternatives. And Dr. Lee's here in the audience, and she actually was involved in some of this work. Jerry Segrist, who many may know is kind of a leader in my field, developed many years ago, came up, Linus Pauling discovered the alpha helix, but Jerry Segrist discovered the amphicaphic helix in the 70s. And what the amphicaphic helix are is an alpha helix, which the residues are arranged so that on one side you have hydrophobic residues, on one side you have polar residues. And this is similar to the structure of the proteins on APOA1. So I'm going to talk about this particular peptide, which is a dimer, and usually the amphicaphic helices have oftentimes have proline in between. Now, I was interested in, or studying these peptides because I studied a disease called Tangier disease, and it was discovered at the NIH by Don Fredrickson, who went on to eventually become director of NIH. And he discovered several cases of a young boy and a sister on Tangier Island in the middle of Chesapeake Bay. And it's a very interesting story. This island was discovered by Captain John Smith in 1608. And there's about 5 families that live on the island. There's no space on the island, it's actually sinking. And there's no room for cemetery, so you bury all your ancestors in your front yard. So this is actually a picture of someone's house. So it's a great place to do genetic studies. And it's essentially a storage disease which is stored as cholesterol ester, particularly in macrophages. But you do have it in the cornea. You get what the question that I often ask medical students is the main clinical finding is you get these enlarged tonsils. They have this orange appearance because they have fat, mostly beta-carotene. They get lymphadenopathy, hepatosplenomegaly, again, mostly from macrophages, but they also have accumulation of Schwann cells of cholesterol where they develop peripheral neuropathy. And our lab and several labs discovered back in 1998, 99, that the mutation was due to what I showed you earlier, the ABC transporter. There's about 45, 50 ABC transporters. They use ATP and they flip various things across the membrane, including lipids. And so the original patients that Don Fredrickson found had 2 base pair deletion, causing a truncation. And then what I found is that if you transfect cells with the ABC transporter, it confers the ability of the cells to efflux or pump cholesterol. The way this assay works is these are helo cells, you add radioactive cholesterol, and in the media, you add either HDL or APOA1 to make protein component, and you monitor the ability of the cells to egress or pump out the cholesterol. So you can see here in yellow, after you transfect the cell with ABC transporter effluxes cholesterol, and it's been previously shown about 5 years earlier from skin fibroblasts that these patients are defective in this process. And then what we find is that this is true not only for APOA1, but it's true for all these other apoproteins, which don't have a lot of primary acid homology, but they all have an aliphatic helix. So we were, at that time, thinking that this was the key motif that was necessary for this process to occur. So I went and used the peptides developed by Jerry Segrist and explored their ability to efflux cholesterol by the transporter. So this is APOA1, showing that if you transfect the cells, they can efflux cholesterol if they don't have ABC when they don't. And if you take the original peptide that he made, it actually works quite well, but it actually worked to a fair degree by the control cells. And you'll see later that the peptide is toxic, and it's too good of a detergent. It's a protein detergent, and it's solubilizing lipid microdomains besides the one created by ABC1. Now, if you take that same peptide and you intermix L and D amino acids, that prevents their ability to form a helix. And so you have the same sequence, but it's not helical, and then in that case the peptide is inactive. So we knew from this work that you needed an amphipathic helix, and it also didn't matter whether it was made with D or L amino acids, you just simply need an amphipathic helix for this process to work. So we were interested in trying to develop a non-cytotoxic mimetic peptide that was specific for efflux cholesterol of ABC1, and that might be safer and more cost-effective than the full-length protein. So this is the peptide that Jerry Segrist developed, and when you look at the natural helices in ApoA1, they tend to have residues in the wrong position. They're not perfect amphipathic helices. It's only the terminal helices that have very high lipid affinity. So we made a bihelical peptide where we purposely took one of the helices and we took these hydrophobic residues and we made them change them to alanine. So alanine is only partially hydrophobic, so it reduces its lipid affinity, and I thought it would perhaps reduce its cytotoxicity. And we called it the 5A peptide. And we made a series of these peptides with either 1, 2, or up to 5 substitutions in the second helix, or substitutions in both helices. So as I mentioned, the parent peptide is very hemolytic or cytotoxic. It lysed about 30% of red cells after about an hour. But making just a few substitutions in the second helix, and by the time you made 5, it was no longer cytotoxic or hemolytic. Even more interesting, the parent peptide causes non-specific cholesterol efflux because it actually can extract cholesterol from the plasma membrane independent of ABC1. But as you made these substitutions, it became more and more specific, and the green represents ABC1 cholesterol efflux. So this 5A peptide with 5 substitutions in the second helix acted much like the full-length protein. It largely only can remove cholesterol from cells expressing ABC1 and very little from the non-transfected cells. Now, if you made substitutions in both helices, the peptide was inactive in cholesterol efflux. So you needed one helix that would have to be very high affinity and one low affinity helix. So we went on to build structural models of the peptide. We have a prolink between the two helices, and so it actually comes back and folds upon itself when it's in lipid-free state, and most of the hydrophobic residues are in between, kind of protected from the aqueous environment, except for two amino acids right in this hinge region. So we have this kind of pitchfork kind of model that we think that these two residues are very important initial insertion into the membrane. And sure enough, if you take these two residues and you change them into more polar residues, it does an efflux, and we found a very close correlation between cholesterol efflux and phospholipid binding. So this is our model of how we think these peptides work. So the original peptide does remove cholesterol by the ADC transporter. ADC transporter flips lipids, phospholipids, primarily phosphatidylserine, to the outer surface to kind of relieve the membrane strain. You actually get evaginations in the membrane, and you decrease the packing of the head groups between the phospholipids, and it allows the natural proteins or these peptides to bind. And then they strip off phospholipid and some cholesterol and form these nascent particles. Now, if you have too good of a protein detergent, it actually will access sites besides ABC1, and which leads to cytotoxicity. But by reducing lipid affinity of the second helix, it's now specific for ABC transporter, and you needed these two residues to actually cause initial insertion. So we went on then to see how this peptide would work compared to ApoA1 in mouse models. So if you take this peptide and you add a phospholipid, it forms these discoidal structures that kind of stack by electron microscopy. And if you inject these into mice, it mobilizes about 5% of total body cholesterol in about 2 or 3 hours, and basically acts as a sponge and mobilizes cholesterol from everywhere, including the liver, but hopefully also from macrophages. So this is very similar to what you would see if you infuse HDL into a patient or into a mouse. And we did what's called a global reverse cholesterol transport assays, and a new method developed by Kinemed. It's a stable isotope dilution assay. It allows you to monitor the flux of cholesterol from the periphery. And we compare that to ApoA1 versus the 5A peptide. And you can see that both the ApoA1 and the peptide increased the mobilization by about 40 to 50% at this dose. In this experiment, what we did is we took macrophages, which is the cell of interest in the atherosclerotic plaque. We labeled them with tritium cholesterol, implanted them into the peritoneum, and then injected the mice with the peptide, and we're able to see that there was an increased amount of counts occurring in the plasma and increased fecal excretion of cholesterol in the stool, which is what we want to occur. And then in collaboration with Phil Barber, we used a rabbit collar injury model We put a celastic collar around the carotid of these rapids, and it causes inflammation. You get VCAM1, ICAM1, you get infiltration of inflammatory cells. And you can see that the 5A peptide responded similar to apoA1 in dampening inflammation from this model. This is some work I did recently with a colleague at NIH, Stu Levine, and Stu is a pulmonologist. And some of you may know that there's a lot still to be learned about HDL, and it has very potent anti-inflammatory effects, which we don't fully understand yet. And so Stu tested the 5A peptide in this mouse model of asthma that he induced with house dust-like allergens. And the peptide actually worked better than steroids in suppressing inflammatory cells, cytokines, and improving airway resistance. So we think this may be related to the cholesterol E-flexibility of the peptide, but there may be other properties of HDL that account for these anti-inflammatory effects that we still don't understand. Now, when you take the peptide and you treat ApoE knockout mice several times a week for about a month, we get a rapid decrease in atherosclerotic plaque, upwards of about 50%, very similar to what you see if you really study the studies in done in humans. Now, before I talk about LCAT, just a few more things about the peptide. So we're very excited about the peptides, but it still has to be used as a acute treatment. And there's a lot of interest now, mostly in the world of vaccines. Many of the antigens for vaccines are helical regions on the surface of proteins. But a protein which may be, a segment which may be helical in the full-length protein, oftentimes you synthesize the peptide, it's just a random coil. That's because the water competes with the hydrogen bonding. And there's a guy named Greg Verdine discovered if you use a hydrocarbon staple, you can actually lock it into this conformation. So this is a movie you can't see. But so we've used this trick, and we put the hydrocarbon staple on the hydrophobic side of the amphipathic helix. And we did this for the last helix of apoA1, which doesn't efflux very well. But if you staple it, it now becomes almost 80–90% helical, and actually effluxes much better than our parent peptide 5A. And we've been able to put these peptides in water and been able to reduce atherosclerosis in ApoE knockout mice. So we licensed the peptide to Kinemed Inc. as a company in California, and we're close to completing our tox study. So if everything goes well, we hope to do an IND and test the peptide at the NIH sometime in the next year. Now, we've made further progress on LCAT and completed a clinical trial. So I'm going to tell you about the background of LCAT and then show you the results of the clinical trial. So LCAT is a plasma protein. It's relatively small, about 60 kilodaltons, 400 residues. It's heavily glycosylated, and it's mostly made in the liver, although it's made to a small degree in the brain and other tissues. And it's mostly bound to HDL, and it's a very low protein concentration. So one of my kind of informal mentors at NIH is Roscoe Grady, and Roscoe developed this idea of using glucocerebrosidase for treatment of Gaucher's disease. So Roscoe was telling me, Alan, why don't you make a private LCAT for LCAT deficiency? So the fact that it works in the plasma, it doesn't require any kind of cofactors, makes it a good target. So that's how we started this project. And LCAT has two basic enzyme activities. It has a phospholipase A2 activity. It clips acyl chains off of phosphatidylcholine and other phospholipids. Then it transesterifies it or attaches it through an ester bond to the hydroxyl group of cholesterol and produces cholesterol ester. So this is the only polar part of the cholesterol molecule. So this enables it to be an amphipathic surface lipid. So when cholesterol is on this bilayer structure, it's on the surface with other phospholipids. But once LCAT esterifies it, it becomes very hydrophobic and partitions from the surface into the core, and that makes this spherical HDL. Now, it turns out it's very important in HDL formation. These are very small and they're very quickly catabolized. So if you don't have LCAT, you can't produce the spherical HDL and you have no HDL. So these patients, like ABC1 mutations, have very low or undetectable HDL. And this again reminds you where it fits in the RCT pathway. It esterifies the cholesterol, and the esterification helps drive this reaction, and about 2 grams of cholesterol are removed each day through the process of esterification. Now, this is a very complicated kinetic model that was developed by Chuck Schwartz, who looked at trace cholesterol and cholesterol ester in humans. And what he found from this study is about approximately 2/3 of cholesterol that return to liver returns to as an ester after being made by LCAT, and the majority of which are on ApoB-containing particles. And that's why, as said earlier, inhibition of CETP is controversial. 1/3 goes directly on HDL, mostly as free cholesterol, and about 90% of the esters that are made are formed by LCAT that they return to liver. So once it's esterified, it's almost a one-way street in terms of returning it to liver. And the body mobilizes about 8 grams of cholesterol a day, but only about 2 grams of that are esterified, suggesting that this may be a rate-limiting step. A lot of the cholesterol that initially effluxes can go backwards. Cholesterol can rapidly exchange, but once it's esterified, you drive this reaction. So, as I mentioned, we were interested in treating patients with familial LCAT deficiency. I'll just show you what that disease is. It's, I think, very straightforward. But we also had the idea to use it acute coronary syndrome. Familial LCAT deficiency is extremely rare, so the reason it hasn't been done before was no profit, no incentive. But I convinced, at the time, Alpha Corp Pharma, that this could be an application, so they bought the license and produced a recombinant enzyme that I'll show you. So the one question they often ask medical students about LCAT deficiency is the eyes, and tangerines, tonsils. So this is the eye of a patient with LCAT deficiency. It's sometimes called fish eye. So Tangier patients can have cloudy corneas, but not usually to this degree. These patients also, it looks like it has arcus senilis, but it's also cholesterol deposit. And surprisingly, it doesn't interfere with vision too badly, but many of these patients do need corneal transplants eventually. It's a rare autosomal recessive disease. They develop anemia, which is usually modest, but it can become quite severe. And the main problem they have is end-stage renal disease, and I'll explain why in a second. So if you have partial deficiency with at least 10% enzyme activity, you just have the eye manifestations, and it's not that big of a problem. But if you have complete deficiency, FLD, familial LCAT deficiency, you get the renal disease, usually in their 30s and 40s. So these patients have very low HDL, usually undetectable because LCAT is important in a setting of maturing HDL. Unlike Tangier disease, they do have some ApoA1, it is much reduced, but the ApoA1 is on the abnormal particles that accumulate in these patients. Normally, about 75% of cholesterol in the plasma is esterified, but these patients are only about 25%. And this is due to another enzyme called ACAP, an intracellular enzyme. They have very low LDL because a lot of cholesterol on LDL is transferred from HDL, and this part protects them somewhat against developing cardiovascular disease. They have a variable increase in triglycerides, and they have very high concentrations of this abnormal particle called LPX, which I'll explain in a second. Now, many of our patients are actually Mennonites from Oklahoma. And the first patient came, it's quite an interesting story. They were farmers and they came with their traditional clothing, and we were asking how they came to diagnosis. And the husband who had the disease kind of laughed and looked at his wife and says, "Well, my wife diagnosed me when we were dating, and she was looking into my eyes the first kiss." And I was like, because most ophthalmologists don't know about fish eye disease. It turns out she was married to his third cousin who had died of LCAT deficient from renal disease. So based on that one family, there's a whole slew of patients, unfortunately, who have LCAT deficiency in Oklahoma that we now treat. So this is a 2D gel electrophoresis of HDL, where you separate by charge and then by size. This is a normal HDL. HDL has about 16 different species of HDL. This is the discoidal pre-beta, and the alpha-4 is also the large discoidal. So if you're missing LCAT, you can see that you have the discoidal structures here, but you don't have the larger species, and that's what LCAT does. Now, what's somewhat unique in LCAT deficiency is they form these abnormal particles, the LPX. So most lipoproteins are micelles. We have a single layer of phospholipid, and then you have hydrophobic lipids inside. Now, if you don't have any neutral lipids, because you're not making cholesterol ester, you instead form these vesicular bilayer structures or multilamellar structures that are called LPX. Now, the only other disease that these accumulate is patients with cholestasis. In some cases of patients with cholestasis, bile has a lot of phospholipid and cholesterol, but no cholesterol ester. So if you have regurgitation of bile into the plasma compartment, the bile salts are very quickly removed and you form these LPX particles. So they have essentially no apoproteins, maybe a little bit on the surface, no ApoB. They have some albumin trapped, and they're variable in size. And what happens is these particles They have unusual charge. It can be positively charged, and the basal membrane glomerulus is negatively charged. They get trapped there. They're eaten up by mesangial cells, macrophage-like cells, and they form these foam cells like you see in atherosclerotic plaque. And then these cause inflammation. You have a breakdown of the basal membrane. And they usually develop proteinuria in their teenage years that goes on to develop chronic syndrome and end-stage renal disease by the time they're usually about 40. So in the lab, we now make LPX particles by taking bile and mixing it with plasma and dialyzing bile salts, or just simply sonicating. So you can see by electron microscopy these double-track layers. These are multilamellar, ugly-looking kind of structures. And if you feed them to mesangial cells, they're very quickly taken up, and they go into the lysosome. So we were interested in what's causing the damage, the kidney damage. And as you may know, there's a lot of interest now in the inflammasome. The inflammasome is a kind of a part of the innate immune system. And anything that causes lysosomal accumulation oftentimes interferes with lysosomal integrity. And you have a release of cathepsins that then cleave caspase that then generate IL-1 beta. And IL-1 beta produces MCP-1 and then produces TGF-beta and causes fibrosis. So what we found is that taking these mesangial cells and treating them with LPX actually causes increased lysosomal stability, and it stimulates IL-1 beta production. And quite interestingly, if you pre-treat the cells with HDL to remove the cholesterol from the cells, or cyclodextrin, which is an artificial substance that's used to complex the drugs, but it does a very good job of removing cholesterol, it actually diminishes this response. So we think patients with LCAT deficiency have a double whammy. They have these LPX particles that go into the lysosome. I think because they're multilamellar, they're very hard to digest. It looks like a type of phospholipidosis, like you would see like with sphingolipidosis. And they accumulate and they cause lysosomal damage, and this is what part causes the disease. Now, as I mentioned, I convinced Alpha Pharma to do this because we thought that it might be useful for acute coronary syndrome. Instead of using HDL therapy, we thought we might be able to achieve the same thing by infusing recombinant LCAT. Easier than infusing HDL itself, or it could potentially be used in conjunction with HDL therapy. Now, it's been somewhat controversial whether LCAT is good or bad. As I said, they have very low HDL, but they don't seem to have very striking increased heart disease, and that may be because they have low LDL. This was a study I did a few years ago where we had samples from the Copenhagen City Heart Study, patients with high HDL, greater than 60, or low HDL, and they either had heart disease in red or not. And what we found is that patients with heart disease tend to have higher pre-beta HDL, the discordant structure. And this has actually been shown now in many other studies that pre-beta HDL is not a bad particle, but if it accumulates, you probably have a defect in the RCT pathway. One defect could be mutations with APC1 causing increased pre-beta, but the other one is low LCAT. And indeed, we found that many of the patients that had high pre-beta had lower LCAT. And we found an inverse relationship in patients with cardiovascular disease between increasing pre-beta and lower LCAT. We didn't see a relationship in normal individuals. And then the best studies looking at their cardiovascular risk were done just recently by John Castellan using three-dimensional MRI and carotid, and they found that they had about a 30-fold increase atherosclerotic plaque. So it's not like FH, but they do have increased cardiovascular disease. So we, as said, developed a relationship with Alpha Pharma, and they made recombinant LCAT for our studies. Initially in HEC cells and most recently in CHO cells. We added it to red cells to look at cholesterol efflux. So this is passive efflux, there's no ABC transporters. So if you add labeled red cells, add radioactive cholesterol, you add CSL-111, which is one of the clinical preparations that recombinant HDL has been used in clinical trials. It can remove cholesterol from cells, but if you do it, if you add LCAT present, you get much more efflux. Whereas LCAT by itself will not work. So again, it shows that the HDL works synergistically with LCAT removing cholesterol. And if you label lipoproteins with either free cholesterol or cholesterol ester, the free cholesterol can go backwards back to the red cell. But once it's esterified, you see much reduced. So LCAT helps in both parts of this efflux pathway. So we made LCAT knockout mice, and the LCAT knockout mice look very much like our humans. So normally mice have mostly, this is gel filtration profile, most of their cholesterol and HDL, but if you knock out the LCAT gene, they have very low HDL, they have increasing LDL, and they have these abnormal LPX particles. And if you add the recombinant enzyme, we were very surprised. We saw a dramatic effect shown here within 1 hour. The LPX particles kind of dissolved, and you were able to make HDL. And this effect persists for a couple of days in the mice. So we weren't anticipating this. We were thinking that the recombinant LCAT would prevent future formation of LPX, but it looked like it was acting on pre-existing particles. So what we did here is we took a patient with plasma with LCAT deficiency. You can see before adding the LCAT, very low cholesterol ester, but adding LCAT for just 1 hour, you esterified to almost normal degrees of esterification of cholesterol ester, and you actually make HDL. It turns out that what residual ApoA they have is on these abnormal particles, and once they acquire cholesterol ester, they can spontaneously reform HDL-like structures. And this was shown here by electrophoresis. So most of the studies we've done to date, we've done intravenous infusion, but so far we've, in animals, have found that you can deliver it IM or sub-Q. It seems to get there, gets the plasma. And we've recently made a slow release preparation of LCAT using a biodegraded polymer that we're going to try in the mice, and hopefully we hope that we'll be able to treat our patients. We think probably once a week subcutaneous administration might be enough to prevent the biochemical abnormalities in these patients. When we infuse the LCAT in our mice, we see modest, this is 4 treatments every day for a 4-day period, modest decrease in cholesterol in the aorta, the spleen, and kidney, but we see remarkable reductions in cholesterol ester. So when cells have excess cholesterol, they store as cholesterol ester. So you can see that these cells are partly starved of cholesterol because the LCAT potentiating the cholesterol efflux. We did not mobilize as much cholesterol ester from the aorta of these mice because I think a lot of the lipid is actually extracellular at this point. It doesn't get rapidly removed by LCAT. And then when you look at gene expression changes of the LCAT treated mice, it's very consistent with the cells are cholesterol starved. They have induction of the LDL receptor, induction of H-amine pyrureductase, DRUB-F2, a transcription factor that turns on cholesterol biosynthesis. And the genes, the transporters that efflux cholesterol that are induced when cells are cholesterol loaded actually go down. Now this is the clinical grade reconstituted HDL that's been infused in patients. I think I forgot to mention, but HDL is not only the substrate for LCAT, but also ApoA activates LCAT. So if you add HDL to plasma, it increases the amount of cholesterol esters that are made. If you add the enzyme to human plasma, you get more esters, showing that the LCAT is rate limiting to some degree. But if you add the two together, you get this nice synergy, suggesting that they may work together. So we tested that in mice, where we infused the CSL-111, which is ApoA combined with phospholipid with or without LCAT. Here we're just looking at free cholesterol, didn't see a difference. But only if you have extra LCAT on board, you get cholesterol esters being formed. And if you do this in the LCAT transgenic mice, all the cholesterol that's mobilized is esterified. And you get more total cholesterol that's removed. So we think that the LCAT could be used alone or in conjunction with HDL, potentially the ability of HDL to remove cholesterol from the body. So we got an IND approved late last year to do a phase 1 clinical trial with our recombinant LCAT. It was a single dose escalation study that we used 0.9 mg, which is kind of replacement level, and we went much higher than that. It was a 1 hour infusion, and then we watched the patients for several days, up to about a month follow-up. And because this is a phase 1 study, the primary endpoint, of course, was safety, but at this study, you'll see in a second, we got very interesting pharmacokinetic and pharmacodynamic data related to LCAT and HDL. So we recruited patients that had stable coronary artery disease, either had a MI or a revascularization procedure between age 30-85, BMI 18-35, and we tried to get people who had normal or normal to low HDL patients. So we ended up with 16 subjects, 14 males, 2 females, 12 Caucasians, and 4 other races. The mean age was 67, and their HDL was on the low side, a little bit under 40 mg per deciliter, so the normal range is about 40 to 60. These patients were stable, but they had lots of other comorbid conditions. They oftentimes had hypertension, diabetes, dyslipidemia. They were on an average of 7 drugs, and most importantly, almost all of them were on statins, and several of them were on niacin. So all the subjects were able to complete the study. We had no serious adverse events. We had no infusion reactions. We had no clinically significant changes in laboratory test results or vital signs or physical exam findings. 2 patients, however, developed a rash, many times several days after infusion. They spontaneously resolved, so we don't know if it was due to the drug or not, but it could possibly have been due to the infusion. So this shows the effect of the infusion on HDL cholesterol. So this is the delta, the change in HDL cholesterol. So at the low dose, which is replacement level, we didn't see much of an effect. But at the 3 mg per kg dose, we got about a 5 mg per deciliter increase. And it seemed to plateau at about 9 mg per kg, where we had on average about a little under a 15 mg per deciliter increase. So we saw nearly a 50% increase in HDL cholesterol in these patients. And the cholesterol ester showed a very similar course. It peaked usually like on day 1, and it had a slow decline over about a week period. This was an IV infusion, so we got the peak LCAT mass at day 1. So this is ELISA LCAT. This is activity. So the one thing I was disappointed about the study is that based on the literature, when patients with LCAT deficiency have been infused with plasma, it's been described that they correct their abnormal lipoproteins for about a week or 2. So we thought LCAT would have a very long half-life. But I think what happens is LCAT jumps on and off. There's only one LCAT molecule for every 100 HDL, so when it falls off, it's very quickly catabolized. So the effect, the prolonged effect you see is due to the initial esters that are made very acutely in the first day of infusion. And it takes about a week for the cholesterol esters to be cleared. So this is the effect of alkaline infusion on the apoA1, which is the main protein in HDL. It peaks actually on day 2 or day 3, and actually you get sustained elevations for almost the whole week. We really see too much of a change in triglycerides. Now, we didn't anticipate this, and we still don't understand why, but we see a very transient drop in LDL cholesterol after infusion. Then it actually goes up. And this is actually better, easier to show here. So HDL peaks at about day 1, then it comes down, and then LDL cholesterol goes up and peaks at day 3 or so. So what we think is happening is that we're esterifying cholesterol in HDL, and then it's transferring that cholesterol to by CETP to LDL, and then the LDL is delivered to the liver. So this is, we were a little alarmed. We saw LDL going up because we don't want to raise LDL, but we think this is part of the normal pathway. So this is a non-donation gradient gel of HDL, and it's just separating by different sizes. So this is baseline patient, and you can see this is the discoidal structure here, and the alpha-4 is discoidal. And when you add the LCAT, the pre-beta disappears because they esterifies the cholesterol pre-beta, and they mature into these. So this is the alpha-4. It goes down to a smaller degree, but you make these larger larger spherical HDL structures. This is plasma. If you take plasma from the mice, from the humans treated with LCAT, and you measure the ability of the plasma to efflux cholesterol, it actually goes down initially. This is ABC1 efflux. This is because the pre-beta HDL disappears, but then when it comes back, it goes back higher levels. You have more HDL, and you get more efflux at later time points. Now here, what we did is we compared the change in HDL that you observe the LCAT in orange versus the 2 clinical trials using the CSL-111, which is ApoA1 purified from outdated plasma. It's a company in Australia. And Serenus is a recombinant ApoA1, actually in Michigan, Ann Arbor. And you can see if you infuse the 2 HDL preparations, you get a rapid rise in HDL due to the ability of the HDL to pull cholesterol from cells. It takes longer for LCAT, but LCAT actually you get a higher rise in HDL, presumably more cholesterol mobilized. And if you look at the amount of esters being made, the Serenus material, probably because the lipid that it's combined with doesn't, it actually inhibits LCAT. Most of the cholesterol mobilized stays as free. With the CSL-111, you get some ester, but with the LCAT infusion, you get much more ester. And if esterification is important in driving net efflux, we think this will be beneficial. So we've treated only one patient so far with LCAT deficiency. This is a 54-year-old man who had very severe renal disease, had creatinine of about 6. He also had very severe anemia of about 7, despite being treated with Lipoprotein. And this is his 2D gel. So he has the pre-beta, the alpha-4 dyskoidal, and has very little of the mature HDL. But after treating with recombinant LCAT, he now makes the full complement of HDL, and we're able to get his HDL almost back to normal levels, and it persists for about a week. As I mentioned, he had very bad renal failure. I don't know to what degree it's going to be reversible, but we saw some acute benefit. I think this may be all we're going to get in terms of treatment, but hopefully we're going to halt progression, and we think we can start treating these patients when they're younger, before they develop any kind of renal disease. This would be the best solution. But he loves us, he loves the treatment, mostly because not his renal function, his anemia. He has, one point his anemia was, he had a hemoglobin of 4, and actually had to call the emergency room. But he normally lives around 7, and he could not walk up steps. He was very incapacitated. His hemoglobin now is like 11. And so they have these cholesterol accumulates in the red cells. So we think, I didn't anticipate this, but I think we're helping his anemia, probably more so than his renal function. So in summary, ACP-501, which we call recombinant LCAT, was safe and well tolerated. We had no serious events. We had 2 possible adverse events from mild skin rashes. No clinically significant changes in lab tests. The LCAT mass activity increased with dose and returned to baseline by 48 hours. We think we have evidence that the reverse cholesterol transport pathway was stimulated. We saw a rapid increase in HDL, increased esterification, increased cholesterol efflux by plasma. We saw this drop in LDL followed by a rise, which we think is CETP. And we think this data supports its safe use in future studies. And about a month ago, AstraZeneca bought Alpha Pharma. And so we're working with them to look at other clinical trials with recombinant LCAT. So I mentioned Don Fredrickson. So Don Fredrickson discovered Tangier disease. In his first paper, he wrote this. So I paraphrased by adding LCAT. But it took about 50 years for this to happen. But I think we are starting to use these rare patients to understand pathways and come up with potential new therapies. So this is a picture of Tangier Island in the Chesapeake Bay. And these are some of the people in my lab, some of which have left, but who worked on LCAT. Bob Schoenberg is a great clinician who actually did the clinical trial. And then we worked with Alpha Pharma, which was part of AstraZeneca. And the peptide work was done in collaboration with Scott Turner from Kinemed. So I'm happy to answer any questions.
Questions for Dr.
Yeah, when you administered LCAT, you saw that transient change in LDL, and the LDL kind of bounced up afterwards. Some of your patients were, and I think some were not on statins. Was there any differential response of those that were taking the statin versus those not?
Yeah, I haven't looked myself, but I don't believe so. But we need to look at that more carefully. And likewise, I think we need to look also at the patients on ice and see if there's some response. So I don't know the answer.
Thanks.
Alan, really very interesting, several different aspects of the study. So 2 questions. So the one about your peptide 5A.
Right.
So as you know, that the 4F somehow, when they did the D, the D amino acid,
Right.
and oral administration appeared to be more effective than injection. So I was just wondering, so in your peptide, have you done the oral one, so how that compares with the other peptide?
Right. Yeah, so this idea of using the peptides is not unique to me. Anantha and Jerry Segrist developed the D4F peptide, which is a single helical peptide made with D amino acids, and Novartis did a clinical trial on that. And so the D peptide was halted because it was available orally, but the D peptides, D proteins aren't degraded, so they were getting long-term tissue accumulation. So they switched to the intravenous preparation called L4F, and it didn't work. And they have a recent paper, Dr. Lee referred to, that came out where they think the reason these drugs work is that is the route of administration through the intestine may be very important, and that these amphipathic peptides may be sequestering oxidized lipids and produce something in the intestine. So I haven't tested my peptide. We have made these staple peptides, which are orally available, which I designed thinking they needed to get into the circulation, but maybe they were working at the level of the gut. So I don't know.
Okay. So the second question, the second part, the LCAT, the recombinant LCAT experiment looks very promising. So is there any studies on the small molecules for LCAT activators?
Right.
Would that be, because you're dealing with protein in this case, so stability is an issue and all that.
Yeah, so Amgen had an effort to find LCAT activators. I think the program, they kind of lost their nerve and were focusing on other things. But they identified a small molecule that activates LCAT quite dramatically, about 2 to 3-fold. And so we synthesized that compound, and in our hands we've been doing it, and it works quite well. And quite interestingly, the patient that we've been treating, most of the mutations in LCAT there's a catalytic triad or usually elsewhere. His esterification goes up quite dramatically on the drug. So we are very interested in that. And what's believed is that LCAT has a hydrophobic lid that covers the active site. And there's 2 free cysteines. So I think this drug is reacting with one of the free cysteines and is locking the lid in an open position. So yeah, so we are very interested in that, and I'm working with some people on other drugs because I think long term, it would be great if we can have small molecule for acting all that.
Right. Thank you.
It's hard work.
Yeah. Thank you.
As we keep cirrhotics alive longer, one of a lot of the complications we see in end-stage cirrhosis are attributed to loss of LCAT. Has anyone looked at that population?
A lot of patients with liver disease have low LCAT because the LCAT's made, and so when they start having compromise in their synthetic ability, the LCAT goes down, the HDL goes down. I'm not aware of, I haven't thought about that in terms of whether they would benefit, but one of the things we are interested in is patients with chronic kidney disease, not from LCAT deficient, just chronic kidney disease in general. They have very low HDL, they have very low LCAT. Those patients oftentimes don't respond very well to statins. They have a very high annual mortality of about 25% from cardiovascular disease. So one of the things I'm talking to AstraZeneca about is whether patients who have come in on dialysis, whether we give them some LCAT might help them. But so we're talking about that, but I haven't thought about the cirrhosis and liver disease. It's interesting.
The clinical trial from that, did you say cellular samples and have you looked at those yet? See whether you're getting changes within the cellular component?
Yeah, so the only thing we've done so far is we looked at the red cells, and the patient with LCAT deficiency, the red cell cholesterol goes down and it normalizes his phospholipids. But from the phase 1 study, we didn't look at that. As I mentioned, there is a technique using stable isotope dilution to measure efflux of cholesterol. So that's one of the things we propose to do in the future. It's very hard in mice. The commonly used animal model is to take macrophages and plant them in the peritoneum. But those kind of studies are, of course, difficult in humans. And I think doing skin biopsies or things isn't probably a useful approach. There's great interest and need in trying to develop imaging studies to actually look at cholesterol flux in the body, but those tools aren't available at this time.
Any other questions?
Hi, Dr. Martin. Very interesting talk, very impressive. I have 2 questions. The first one, you mentioned that you were disappointed to see the short half-life of LCAT. So I wonder, have you looked into the catabolism pathway of this enzyme, just like as you did for those HDL peptide, maybe manipulating some of the peptide can increase the half-life?
Yeah, no, we haven't, but you're right. There's a lot of interest in pegylating proteins and doing various modifications, cytokines, and something that we might be able to do in the case of LCAT. Because otherwise, I think these patients will need, right, at the present time, will need to get treated at least once a week, intravenously, which is very difficult. Although a lot of the lysosomal storage diseases, that's the frequency they're treated. So we are thinking about ways to extend the half-life, although that may, there's about 1 LCAT for every 100 HDL, so it has to move on and off. So I'm afraid that maybe pegylating might affect that process, but we are interested in that. And as I mentioned, we're also trying to make a slow release preparation. LCAT is it's very stable enzyme, even on the bench. So we think that if we can make a slow release preparation, that it would maintains activity and release over a long period of time might be another way to handle the problem.
And just a wild thought, since it's a recombinant protein, is it possible to make an inducible form?
What?
An inducible form, like you can just infuse a non-active form in the circulation and then maybe make it inducible with some activities.
Yeah, maybe. Yeah.
Another question that I have, I'm just curious that these patients getting this infusion of HDL or LCAT, are there, like in the mouse model, did they look at the liver, that these mouse, are the liver readily handle this extra load of cholesterol that is just brought back to the liver?
Yeah, so it turns out that most of the cholesterol that goes up afterwards actually is not necessarily from the periphery, it's actually from the liver. Since the liver has an open sinusoidal system, if you do anything to increase the ability of HDL to extract cholesterol, it acts on the liver. So it's kind of like a reverse. It pulls it from the liver, partly pulls it elsewhere, and then eventually all goes back to the liver. At the very high doses, actually, you get some elevation of transaminases, and they think it may be due to the big load of cholesterol coming back to the liver, but that's just a hypothesis.
Does the 5A peptide cross the blood-brain barrier, and if so, what effect might it have on cholesterol metabolism in the CNS?
Yeah, we haven't looked. It is small enough, and apoA1 does cross. There's likely to be some transporters, and as you may know, there's a lot of interest in apoA1 and Alzheimer's disease. There's been both knockout and transgenic mice of apoA1 showing that increasing apoA1 helps prevent the cerebral angiopathy that accumulates around the vessels of the brain. So it's something that we're interested in. We haven't looked at. Any more questions?
Well, thank you very much.
Yeah.